Hi, welcome to Science Hangout from Science Mission. I'm Sadashiva Pai from Science Mission. Today we have Dr. Suzanne Lapi, Associate Professor of Radiology and Chemistry and Cyclotron Facility Director at the University of Alabama at Birmingham with us. In addition to publishing several articles on peer-reviewed journals and membership in several societies, Dr. Lapi has won several awards to her credit. In 2014, she has won Marie Curie Lecture International Conference on Isotopes. In 2010, she won Mario Nicolini Prize from Italy. In 2010, she won Harry, Harry Gray Family Fund Award, Metals in Medicine in New Hampshire. In 2007, she won President's Research Stipend, Simon Fraser University. In 2006, she won several awards, one of them being student travel bursary to a symposium, chemistry poster competition, student travel, again bursary from Midwinter for attending Midwinter meeting in Arizona. In 2005, she won student again award to attend Pacific Rim Chemistry Symposium and chemistry oral competition. She won second place in SFU. Welcome, Dr. Lapi.
Hello, nice to be here.
Thanks for joining Science Hangout. Good.
Thanks for inviting me.
So good to have you here with us. And can you tell us about yourself, your background, your education, etc.?
Sure. So I started off in chemistry and then became more interested in chemistry with radioactive isotopes. I did a master's degree looking at reactions that happen in stars. So I was really interested in nuclear reactions that happened in supernova. And then I started to transition more to the medical field. So I became interested in how we can use radioisotopes as tools in medicine, and particularly in medical imaging. So I did my Ph.D. at Simon Fraser University looking at isotopes for medicine. And then I did a postdoc at the University of California, San Francisco, where I continued to look at imaging agents, mostly for cancer. And then I was at Washington University in St. Louis as an assistant professor and then a tenured professor. And then in 2015, I moved to the University of Alabama at Birmingham as a tenured associate professor, where I run the Cyclotron facility and develop new molecular imaging agents.
Okay. So can you talk us about your mentors, role models you have come across in your research career?
Sure, I've been very lucky and have had wonderful mentors. So starting with my PhD supervisors, Dr. Tom Ruth and Dr. John Doria, really gave me a love for the field of radiochemistry and how we can use radioactive materials for a whole host of things, including medical imaging. Then my postdoctoral advisor, Henry Van Brocklin, again, a lot of enthusiasm, and really started to learn more about how can we develop radioactive tracers and then translate them into people, and how can we get them more translational bit and more into clinical trials and that sort of thing. Then when I moved to the Washington University in St. Louis, I was fortunate enough to work with Dr. Mike Welch, who again convinced me that you really could have a program where you go all the way from particle accelerators, making radioactive isotopes, to doing the chemistry, to putting all these things together, you know, small animal trials, and then human imaging as well. So really gave me the love for the breadth of the field, and also training and mentoring and having all the students along the way. So I learned a lot from my mentors.
Okay. And I know you are busy with doing all the research as well as maintaining the facility. So what do you do during your, what are your hobbies other than the research?
So I'm a chemist, and actually, like most chemists, we like to cook because it's really just different applied chemistry. So I cook a lot at home. I do a lot of experimenting, I guess. My husband and I like to travel, so travel and see new things, new different areas of the world. So I think those are my two main hobbies.
Okay, and let's talk about your lab people. So, can you please talk about them, like how many grad student postdocs, or like what do they do and all?
So right now I have 4 graduate students.
Hmm.
So 3 of them are in chemistry and 1 is in biochemistry. I
Uh-huh.
have 2 undergraduates and they're both biomedical engineering.
Uh-huh.
I have, I guess, 3 postdoctoral fellows and they're all radiochemists as well. So they're working on new molecular imaging agents.
Hmm.
And then I have 3 PhD level staff scientists that are really sort of the core of my team and really help me run my program. I
Hmm.
also have, we have an excellent pharmacist that works for us and helps us transition some of these compounds into clinical trials. And
Uh-huh.
then we have a number of engineers that help with the cyclotron operations.
Okay. And how do you like? There looks like there is a bunch of people in your organization. So, how do you manage like the time with them,
Sure.
advising them, and all those things?
I meet with everybody once a week, so everybody gets a one-on-one meeting. And
Uh-huh.
then we have a group meeting where we all get together and we make sure everybody knows what's going on, knows the schedule. And then a lot of informal meetings. You know, a lot of really good ideas come out of these hallway conversations or just having
Hmm.
lunch with somebody. And so I try to interact with everybody as much as possible.
Okay. And I have seen some of your recent work, which is pretty interesting. And can you please tell us more about it and start with your presentation if you don't mind?
Sure. So I'll just jump right in then and I'll go
Okay.
ahead. So I should just share my screen now.
Yeah, sure.
Okay. Great. So I'm very happy to be here and I'll talk a little bit about our work. I've titled my presentation From Isotope to Images, and we're going to talk about radioactive materials as tools both in medicine but also some recent work we've done in basic science. So we use isotopes for something called the tracer principle. And so in nuclear medicine and many areas of basic science, we can use these radioactive atoms as tracers. So the idea is that the tracer behaves in a similar way to the components of the system that you're investigating, but you're not altering the system in any fashion. So we're using these tracers to sort of look inside the system, see what's going on, but we're not trying to change it. So it's really diagnostic procedures. And of course, the ultimate concept is that the tracer concentration can be measured. So as an example of this, we look at George de Hevesy. So he was a pioneer in radiochemistry a long time ago. He worked in Manchester in the early 1900s. He worked for Rutherford, Ernest Rutherford, that many people may have heard of. And he suspected that his landlady was serving him recycled food. So he takes the Sunday night roast and he spikes the roast with radium. The radium is used as a tracer, the radium's in the roast, and lo and behold, he finds the radium in Wednesday's hash. So he's using this radium as a tracer to find out where the roast goes. And so building on this, we can kind of expand on this idea and we can make radiopharmaceuticals. So the idea is a radiopharmaceutical is a drug that's labeled with a radioactive isotope to image a biological process or to deliver therapy to a specific disease site. The overall chemical structure determines the biological properties of the drug, where the drug is going to go, how it's going to clear out of the body, what it targets. And the radioisotope or the radionuclide that you're attached to your drug determines the imaging or therapeutic properties. So what I'm showing here, can you see my mouse? Is it okay?
Yeah.
So I have our radioactive isotope, and I've got some kind of linker, and then what I call a pharmacophore. This is the drug that we're talking about. So as an example of this, I can take D-glucose, so this is just glucose as a sugar molecule, and I can tag it with a radioactive fluorine atom. So that's this red fluorine-18 here. And I can essentially make a radioactive sugar molecule that we can call 2-fluoro-2-deoxyglucose, or FDG for short. And I'll come back to that in a minute. So our group is really interested in how do we make these isotopes and how do we separate them so that we can get them in a useful form for chemistry. We work on the radiochemistry for new imaging agents. So how do we attach those radioisotopes to the molecular imaging agent? The characterization of new radiopharmaceuticals. So do they bind to the target that we designed them to bind to? translation into clinical trials, and then we support ongoing clinical trials in molecular imaging as well. So let's go back to the radiopharmaceuticals. So we've got this targeting compound that's labeled with this radionuclide for imaging and therapy. The structure of the compound determines the biological properties, the targeting, the clearance, the different receptor interactions, and the radioactive label that we've attached determines the imaging or therapeutic properties. So we have our targeting moiety, we label it with our radioactive atom, then we have our volunteer here, we inject them with our compound, and it essentially lights up like a beacon telling us where the drug goes. And we look at imaging with something called positron emission tomography. And this is a very sensitive tool, so this is how we measure the radioactivity that's in a person. And it can give us information about the biological and physiological processes in a non-invasive manner. And that's really key. So we're injecting radioactive materials into people, and then we can image them without doing surgery and without taking a biopsy. So how positron emission tomography works, we have this radioactive atom that's here. It emits what we call a positron, so that's this E+ here. The positron is the antimatter of an electron. So in the body, when our antimatter hits our matter, or in our electron, they annihilate. And when they annihilate, they give rise to these two gamma rays. And because of conservation of momentum, these two gamma rays are emitted back to back. So when this happens in a person, these gamma rays are emitted back to back. We can use our detectors to see them, and then we get what's called a line of response. If these two detectors fire at the same time, we know that event happened somewhere along this line. We collect many, many millions of these events, and then we can form a picture to determine where the radioactivity was. So let's go back to glucose. So we can use glucose to look at sugar metabolism. So we take our glucose, we've replaced this hydroxyl group here with this radioactive fluorine atom. This is a very sensitive tool, mostly to look at patients that have cancer. Cancer cells take up a lot of sugar, they're growing very fast, they're what we call hypermetabolic, and so we can essentially get a map of metabolism in the human body. And so this is a woman that was actually scanned at our institution. She has been diagnosed with T-cell lymphoma, and the dark spots are indicating high uptake of, let's see if this works. This isn't gonna work. Okay, high uptake of our FDG. And so you can see normal uptake in the brain, you can see normal excretion through the bladder, and normal excretion through the kidneys here. And unfortunately for this woman, you can see a number of uptake spots that are indicative of different metastatic tumors as the result of this woman's lymphoma. So we're using the FDG to help diagnose this woman. So she underwent several rounds of chemotherapy and then came back for a follow-up scan. So this is showing the follow-up scan. Still, you see normal uptake in the brain, normal excretion through the bladder, and normal excretion through the kidneys. But now you can see the pretty dramatic difference between the metabolism before the treatment and after the treatment. So we can use FDG to follow patients during the course of treatment. So FDG is great, and this is a wonderful way to look at cancer metabolism, but we're interested in a lot more things than just metabolism. So we would like to look at detection of cancer cells, prediction of treatment response, receptor status, oxygenation, microenvironment, and a whole host of parameters that can help us give information to the physician to determine how best to treat the patient. So we really want to tailor treatment that's specific to a patient's cancer. And so we can get different information with different tracers. And so this is just a literature figure shown here. So this is a man that had an FDG scan. So this is, again, showing sugar metabolism. And he's actually had a tumor resected on his scalp. So you can see this little sort of blank spot here. And he's being scanned again to look for residual tumor. Unfortunately, you can see a number of dark spots, both in the spine, also still on the scalp. This is just normal liver and then bladder. But if we scan with another agent, gallium-68 DOTATOC, we can see a number of other sites lighting up, really a lot of sites in the spine and in the hips. And gallium-68 DOTATOX specifically targets a receptor on cancer cells called the somatostatin receptor. And this is really important because there are certain therapies that target the somatostatin receptor. So with this gentleman, we're not only determining where his cancer cells are, but the fact that those cancer cells have somatostatin receptors, and that this man is probably going to be a very good candidate for somatostatin targeted radiotherapy. So now we want to build all of our compounds. We need to think about what kind of isotopes are we going to attach to our molecules. And when we think about those isotopes, we think about the chemistry, the half-life, the different decay properties, the availability of those isotopes, and the purity. And so some of the more common PET isotopes, I'm calling standard PET isotopes, are shown here. And so here I'm showing the nuclear reaction that we use to make those isotopes, as well as the half-life. The half-life is the time it takes for half of those isotopes to decay. So we can make carbon-11 by proton bombardment on nitrogen-14. We call this a P-alpha reaction. Carbon-11 has a half-life of 20 minutes. We can make fluorine-18 by a PN reaction by irradiating oxygen-18. Fluorine-18 has a half-life of 2 hours. Similarly, we can make nitrogen-13 and oxygen-15, which has a half-life of 10 minutes and 2 minutes. So carbon, fluorine, nitrogen, oxygen. These are a very nice toolbox and an excellent place to get started. And we can use our chemistry to attach these radioactive isotopes to numerous small molecules. However, the drawback with some of these isotopes is that they have very short half-lives. So we're losing half of our molecules in the case of carbon-11 in 20 minutes, or half of our molecules in 2 hours. And so your chemistry has to be very fast. And this also limits the biological imaging half-life, or the biological imaging time that you have. Let's say you want to image some kind of process that happens in a patient for several hours or several days. You're probably going to need something different than these isotopes. So our group works a lot with radiometals, and often these radiometals have longer half-lives, so we can use them to probe longer biological processes. We can use these to expand our toolbox, so we get a variety of half-lives and different decay characteristics that are available, and these can be used, some of them can be used for both imaging and targeted therapy. When we're thinking about metals, if you remember your chemistry a little bit, you need to work on the coordination chemistry. So what we want to make sure is that we are basically developing compounds that hold those metals in place so that the metal stays with the targeting agent. So we really want to know where the targeting agent goes and not just where the free metal goes. And so thus we need to really think about stable chelators. So this is our toolbox. This is the chart of the nuclides. And you can think of the toolbox or the chart of the nuclides as an expanded periodic table. So there's about 3,000 known isotopes. We've got protons going here on the y-axis and neutrons going here on the x-axis. So every row is a different element, and every box is a different isotope. And what I'm just showing here is the color-codedness so that we can see the different half-lives with relationship to where they are on the chart of the nuclides. The ones that we're interested in that decay by positron emission are roughly found here. There's several hundred of them, so this is where we go looking for new isotopes to build our radioactive tracers. So if we're looking at radiometals, you can imagine the periodic table. The first row radiometals are found roughly here. And if we zoom in, we can get a better look at what metals are there. So this shows nickel, copper, zinc, and gallium. The black ones are the stable isotopes, and the colored ones are the radioactive isotopes. So we can start to look for isotopes that decay by positron emission and also have reasonable half-lives. So here we've got copper-61, which has a half-life of 3 hours, copper-62, which has a half-life of 10 minutes, which is a little short, copper-64, which has a half-life of 12 hours, gallium-66, which has a half-life of 9 hours, and gallium-68, which has a half-life of about a little over an hour. If we go a little farther up the chart of the nuclides, we can look at second row transition metals. And again, zooming in, we get strontium, yttrium, zirconium, and niobium. And here we see yttrium-86 that has a half-life of 14.7 hours, and zirconium-89 that has a half-life of about 3 days. And I'm going to focus a lot on zirconium-89 moving forward because we've done a lot of work on this isotope before. So how does the cyclotron facility work? So we enable a broad scope of research and cutting-edge patient care, and we have projects that range from isotope production, so how do we make these isotopes, to developing and supplying these new molecular imaging agents for both clinical trials and routine patient care. So again, kind of a broad scope of activities. This is the layout of the cyclotron facility. So you see here this big dark box that's sort of in purple at the bottom. This is actually the cyclotron vault. So this is where the particle accelerator is, and I'll talk a little bit more about that in a minute. Adjacent to the vault, we have this gray area. This is the equipment room. So this is where all the sort of auxiliary equipment to run the cyclotron is. The yellow area is the preclinical radiochemistry lab. So this is a non-human use area that mostly we use for developmental chemistry. This is where a lot of our students work. Along the side here, we have our GMP facility. So this is a clean air. You have to gown up to go in there. This is where we make all the human use radiopharmaceuticals. And you see these boxes here. These boxes are called hot cells, and they're essentially lead shielded areas that we can use to prepare our radioactive materials in a very safe manner. Next to the GMP facility, we have this green area, which is the quality control area. So here we make sure that the radiopharmaceuticals are pure. We make sure that they're the right compound. We make sure that they're sterile, ready and safe for human use injection. So this is the quality control area. And then in sort of pink here, we have our radiopharmacy. So this is where we actually draw up the final dose and we prepare the syringe where we'll get injected into the patient. This is the sort of close-up of the vault here. So this is actual cyclotron here. So the cyclotron, I'll talk a little bit more about that in a minute, has beam lines on both sides. So we use the cyclotron to accelerate protons to actually induce nuclear reactions to make these radioactive isotopes. So this is the cyclotron here. It was made by a company called Advanced Cyclotron Systems. They're located in Vancouver. It accelerates protons to 15 to 24 mega electron volts, so millions of electron volt protons. It has 2 extraction ports on either side and these 4 beam lines. So we can use the cyclotron to irradiate either solid, liquid, or gas targets. So we pick different target materials depending on the radioactive isotopes that we want to make. This is just showing the cyclotron when it's open, and this is the cyclotron when it's closed. So you can see here's one at the beam line on the far side there. And the cyclotron is in a heavily shielded vault for radiation safety. Okay, so how does a cyclotron work? So the general principle is that you have an alternating voltage with a frequency equal to the orbital frequency of the particles that's applied between the Ds. And this helps to accelerate either positive or negative particles. So what does that mean? Essentially, the cyclotron is housed in a big magnet. So here I was just show north and south here. And then there's these 2 electrodes inside the cyclotron. And because of these electrode shape, they're called Ds. So our cyclotron actually accelerates H-. So this is a proton with 2 electrons attached to it. You inject these into the center of the cyclotron, and because this is a charged particle, it's contained in a magnetic field, it starts to move in a circle. So when our H- gets to what we call this gap in between the 2 Ds, we make this D negatively charged and this D positively charged, and our H- gets accelerated across the gap. It starts to move in a larger circle, and then it gets to the next gap. Then when it gets to the next gap, we make this D negative and this D positive, and it gets accelerated again. Now it's moving in a larger circle, gets to the next gap, then we make this D negative, this D positive, it gets a little kick again, and it goes in a bigger and bigger circle. So in this fashion, we can use a small machine to accelerate protons to very high energies. So this is our machine. So you can see here, this is the magnet, and I apologize, the picture's a little on an angle because I was sort of crunched under the machine trying to get a photograph of it. So this is the machine when it's open. This is the top plate, and this is the bottom plate. And so while our Ds are a little bit funny shaped, they're not exactly D-shaped, they're these sort of pi wedges, they serve the same function. So these electrodes are switching polarity in order to get our protons, or our H minus, to accelerate in a circle to faster and faster energies. So once we get our protons accelerated, we need to actually radiate targets to induce the nuclear reaction. So as I mentioned before, our targets can be gases, liquids, or solids. And so we can irradiate a nitrogen-14 gas target. This is the P-alpha reaction to make carbon-11. So we can tame nitrogen-14 gas in essentially a cylindrical tube and irradiate it with our protons. We can irradiate liquid targets. So this is a P-alpha reaction on oxygen-16, that's just as water, to make nitrogen-13. We can have a PN reaction on oxygen-18 to make fluorine-18, and this is an enriched oxygen target. We can also irradiate solid targets. So this is a PN reaction to make copper-64, and then a PN reaction, this is actually, it should be making zirconium-89 on metallic yttrium-89. So this just shows some of our target stations. So this is actually what's at the end of those beam lines. We have a gas target here. So this is that long cylinder that's probably, let's say, 10 or 15 inches long, that contains our high pressure gas to make our carbon-11. These are our two water targets. They both contain about a milliliter of water that we irradiate to make the isotopes. And this is one of our high current solid target stations for irradiating solid targets. This is one of our smaller targets that we call the coin target. So these coins, as you can see here, they're about the size of a quarter. So we can electroplate, or basically deposit different materials on these coins in order to irradiate them to make our different isotopes. And so here I'm just showing, this is yttrium that's sputtered on top of niobium. We can put them in this little coin target. So this is the coin slot here. The proton beam comes from this side, and then this sits at the end of one of those beam lines. And this just shows the actual physical target. So again, the proton beam comes from this side, and this is that coin slot that I pointed out before. This is our high power target station. So this is actually a larger target. So this is about the size, you can think about the size of a cell phone. You can electroplate different materials on here, on this little copper wedge. This plunger here pushes this up into the holder, and then the beam comes in from this side here in order to irradiate the target. And again, this just shows the actual target station. So this is what you see if you go into our vault. I showed the GMP space on the map a little bit before. I just wanted to show what that sort of hallway of hot cells looks like. So you can see here, these are the manipulator arms that we use to manipulate things inside the hot cells so that we're not exposed to radiation. We have different size hot cells. We also have an area that we can draw up doses for human use. The clinical quality control area has different things to ensure that the radiopharmaceuticals are high quality. So we have HPLC, GC, TLC, so a lot of analytical equipment in order to identify our proper radiopharmaceuticals. This just shows a list of the different radiopharmaceuticals that we currently have in use. I'm not going to go over them all, but I just wanted to show that these radiopharmaceuticals can be used for a wide variety of things. So this mGluR5 compound is actually looking at a receptor that's very important in addiction, so we're doing some neurology studies. This FLT looks at proliferation, so how fast cells are growing. We're looking at cardiac blood flow, so we're doing some heart studies here with this N13 compound. I mentioned dotatate, where we're looking at somatostatin receptor status, and a number of other tracers. Again, a toolbox of tracers to look at different parameters to help physicians decide how to treat patients. We also look at a number of different isotopes, so looking at the toolbox of isotopes in order to make these different compounds. So we routinely produce fluorine-18, carbon-11, nitrogen-13, but we also have many research projects looking at development of new isotopes. And in fact, this is what most of my PhD students work on. So looking at, let's say, scandium-44 that has a 4-hour half-life, looking at how do you make targets, how do you radiate them, how do you separate it out and actually get it attached to interesting molecules. And again, I'll focus a little bit on zirconium-89 as some of our examples. So zirconium-89 is a very interesting isotope. It has a half-life of 3 days, and it's really well matched if we want to look at the pharmacokinetics of antibodies. Antibodies are very large proteins. They circulate in the blood for a long period of time, and so we can actually image the distribution of those and see how well they're targeting the tumor, how long they take to clear from the body, and also estimating dosimetry. From a chemistry standpoint, zirconium-89 is also very interesting as it's quite inert to biological systems. We have previously done a lot of work with copper-64. This is very difficult to work with because you have a lot of enzymes and proteins in your body that use copper, and so you have to really work to sequester it from these biological processes. Zirconium-89 is quite inert to biological systems, and so we can make very stable complexes. We're interested in the positron decay, so that's this beta plus here. So it decays by positron decay 22% of the time, so we can use this isotope for PET imaging. Here I'm just showing the decay scheme, so zirconium-89 decays to yttrium. So when we think about producing zirconium-89, we need to think about the probability that these nuclear reactions are going to happen. If we irradiate an yttrium target with a proton beam, how many zirconium-89 atoms are we going to make? And so what I'm showing here is this cross-section. So the cross-section is essentially the probability that a nuclear reaction is going to happen. And here I have the incident energy of the proton beam. So in blue here, you see the yttrium-89 PN reaction to make zirconium-89. So this is the nuclear reaction that we actually want to happen. And you can see it peaks somewhere around 15 MeV. If we go higher than that, you start to see yttrium-89 P2N reaction giving you zirconium-88. Zirconium-88 is a very long-lived contaminant. We don't want it in our yttrium-89 production. So what we have to do is basically moderate the proton beam so that we get the yttrium-89 PN to make zirconium-89, but we don't get the P2N to make zirconium-88. So it's very, you have to be very careful about the incident energy and the nuclear reactions that are taking place while you're irradiating that. So I'm just zooming in here again on the chart of the nuclides. So we're starting with yttrium-89. This is a PN reaction to give you zirconium-89. And we want to avoid that P2N reaction to give you zirconium-88, which has a half-life of 80 days. So what we do is we take our proton beam that starts around 16 MeV, we run it through what we call a degrader. This degrades the energy of the proton beam so that we have a 12.5 MeV proton beam that's irradiating the yttrium target. We purify our zirconium-89 by ion exchange chromatography. So when we're done with the irradiation, we have our zirconium-89. It's in this matrix of yttrium-89. And we dissolve the yttrium-89 in hydrochloric acid, and then we put it through a resin. By changing what we're putting through that resin, we can differentially elute off the yttrium-89 and the zirconium-89. So this is just showing the functionality of the resin. If we load on the dissolved target, then we wash the target in hydrochloric acid, then we wash the column again in water, and this is all to make sure that we get out all that yttrium target material. Then we can elute our radioactivity in 1 molar oxalic acid, and here you can see the profile of the radioactivity elution. So the zirconium-89 stays bound to the column until we elute the column with 1 molar oxalic acid. So because we're working on scaling up our production of zirconium-89 and working on clinical trials with this isotope, we've worked on automated separations. And so this is why I have biomedical engineering students in my lab, is because they work a lot on these automated chemistry systems. So they basically take the chemistry we've developed. This is the dissolution vessel. So this is where we're dissolving that coin target up in hydrochloric acid. And then all remotely, we can move this onto this column here, where we can elute out our yttrium-89 in that water and in hydrochloric acid. And then we can elute our zirconium-89 in the oxalic acid. And this is all controlled by LabVIEW and has been programmed by one of my undergraduate students. So we use zirconium-89 to do imaging with antibodies. Antibodies, as you may know, are these proteins that can be engineered with very high specificity to certain targets on different cells. We combine this with the very high sensitivity of PET imaging, and so we can make molecular imaging agents that are very specific and very sensitive for certain receptors. So antibodies and antibody fragments are very selective targeting agents. And there's been a wide variety of antibody-based therapeutics that have been developed in the last 2 decades. Every time you see, a news release from a radiopharmaceutical company that says something, something mAb, that is an antibody-based therapeutics. And in fact, probably half of the therapeutics coming out of the pipeline today are all antibody-based. So the idea is we can use these antibody imaging techniques to offer the potential of stratifying patients that may benefit from antibody therapy, monitoring the course of these therapies, and also paving the way for the next generation of targeted therapies. So we've done a lot of work on with an antibody that targets something called the human epidermal growth factor 2. We'll call this HER2. So HER2 is a transmembrane receptor, so it basically spans the cell membrane, has a piece that's inside and a piece that's outside. It doesn't have any known natural ligands, and it's amplified in about 20% of invasive breast cancer. Patients that are HER2 positive have increased tumor aggressiveness. They are shown to be resistant to certain types of therapy and typically have increased mortality. So there are several HER2 antibody targeted therapeutics. So the first one is trastuzumab. So this is shown here. Trastuzumab is an antibody that binds to what we call domain 4. This is the domain that's close to the cell membrane, and it works by suppressing the downstream signaling of the HER2 receptor. The other antibody that's available is called pertuzumab. It binds to domain 2, so this is a little bit farther away from the cell membrane, and it works by inhibiting the HER2 dimerization with other receptors. So again, inhibiting the function of the HER2 receptor. There's also a newer compound called TDM1 that is based on trastuzumab, but is linked to a toxin called TDM1. When this is internalized, it essentially releases the toxin, and the toxin interferes with cell growth and proliferation. So there's these 3 targeted therapeutics, and but they only work if the patients are HER2 positive. So we started with trastuzumab. Trastuzumab is an FDA clinically approved antibody. I just show here, this is the package insert. And so the idea is that a trastuzumab imaging agent may be useful for determining dosing strategies and for predicting who may respond well to trastuzumab therapy. How they do this now is they take a biopsy, they'll do staining for looking for the HER2 receptor. But patients' HER2 status can change over the course of therapy, and even if a patient has widespread metastases, you can see cells that have HER2 positivity and HER2 negativity. And so you really want to know who's HER2 positive in order to determine who would be well suited for this type of targeted therapy. So what we wanted to do was develop an imaging agent that was based on trastuzumab. So we have this DFO, so this is our chelator that's going to hang on to our zirconium, and then this NCS functional group. This NCS functional group, basically we can attach it to these free lysine groups on the antibody, and then we can create our antibody chelator conjugate. After we have our antibody chelator conjugate, we can radio label it with our zirconium-89, giving rise to our zirconium-89 chelator, and thus our targeted radiopharmaceutical. And a lot of this was pioneered by a group in the Netherlands. So what we wanted to do first was make sure that our zirconium-89 trastuzumab still targets HER2 positive cells. So what you're looking at here is what we call a PET-CT image. And a CT is essentially just like a 3-dimensional X-ray. So that 3-dimensional X-ray is windowed for the bone here, so you see that in gray. And the PET is the orange here. So in this way, we can get an overlay of the functional imaging, or where the HER2 is, and the anatomy, which is what the CT is showing you. So these are animals that were implanted with HER2 positive cells and HER2 negative cells. So these are the positive, these are the negative. We inject the animals, and then we image them at 24 hours post-injection. 24 hours post-injection, you see high uptake in the HER2 positive cells and low uptake in the HER2 negative. However, at 24 hours post-injection, you still see a lot of background here. So this is in the blood pool, but this is because the antibody has a very long circulation half-life. And you also see some signal coming from the spleen. If we take those same animals, because this is non-invasive imaging, we can image them in 96 hours after injection. Here we see the HER2 positive, here we see the HER2 negative. You can see that a lot of that background has faded out, and we really have very nice tumor delineation and high uptake in the HER2 positive tumor and low uptake in the HER2 negative tumor. We've also done what we call a micrometastasis model. So here we've taken our HER2 positive cells and transfected them with an optical imaging agent. When we inject them in the tail vein, we can get very, very small tumor metastasis. So here's one in the jaw and one in the spine that are optically active. So we know where the cells are. And then we can image this with our Zirconium-89 trastuzumab. And even in these very, very small lesions, so these are about 1 millimeter across, we see high uptake of the Zirconium-89 signal, again, both in this jaw lesion and in this spine lesion. So moving forward with this, we were really happy to partner with Farouk Dadashi at Washington University to move forward with a clinical trial. And so the idea is can we use this Zirconium-89 trastuzumab to identify lesions that were HER2 positive and to identify lesions that were HER2 negative? This is one of our early studies, so I'm just showing here the PET imaging. You see some liver in the background, and you still see some blood pool. This is actually one of our patients that had already had a double mastectomy, so she doesn't have cancer in the breast area. However, you can see this lesion here. This is in the femur, and this is a known HER2 positive lesion. And so we are very excited about this preliminary result. So we move forward. This is going to work. Okay. We move forward. We were imaging patients, again, 2 days and 5 days after injection because we want to see how well we can image as time progresses. So this is a patient at 2 days after injection. You can see still quite a bit of blood pool. So this is the heart here. You can see some uptake in the liver, and then this is excretion through the bowel. But even at 2 days post-injection, you can see significant uptake in the shoulder lesion, in this hip lesion, and in this femur lesion. This again just shows the PET CT, just sort of focusing in on the shoulder lesion here. This is the same patient 5 days after injection. So again, showing the shoulder lesion, hip lesion, and thigh lesion, and showing a little bit that the blood pool clears out at this later time point. This is an interesting study because, so this is the same patient, so you see the shoulder, hip, and thigh lesion. This patient also had a CT scan to look at her, the extent of her cancer. So the CT scan was in April, whereas our scan was in May. This was not identified on CT. However, we did identify it with the PET scan, and in August of this year, this patient again had a CT scan, and now this was identified. So this, in this one anecdotal study, we actually did see it before it was evident on CT. This is one of our other images, again, just showing that we are able to look at primary breast cancer in the breast here. So here you see 2 lesions. This patient actually has two lesions called multicentric primary breast cancer. And this study is continuing on. So we're continuing on both at Washington University, and we're starting a new cohort at University of Alabama at Birmingham as well. And so we're starting this trial, and we'll actually be imaging patients and then looking at the correlations with response to targeted HER2 therapy. So we're really excited to start that study here. I wanted to give another example about how we can use this technique in radio tracers outside of medicine. And so we recently became very interested in a compound called PFAS, or a family of compounds called PFAS. So PFAS are perfluoroalkylated compounds. They are very persistent environmental pollutants. They accumulate both in plants and animals, and they have toxicity possibility both in biological organics. They're very stable compounds, so this means they have very slow breakdown in the environment, and they hang around for a long period of time. These PFAS compounds are widely used in a lot of different products, and typically they're used to make products more resistant to stains, grease, and water, such as nonstick cookware, stain resistance in carpets and furniture, waterproofing, mattresses, and also in firefighting materials. So some of the PFAS compounds, such as PFOS, is no longer manufacturing, and they are phasing out some of these, but there are other derivatives that can still do harm, and these compounds, again, still hang around in the environment for a long time. So I'm just showing here, these are some of the different compounds in this family of PFAS. So PFAS can be found in water supplies from long-term contamination, partly from firefighting foam. They're also found in fast food materials, wrappers, other materials, and they're known to bioaccumulate. And there's also been several reports of environmental contamination from spills. And so this is a lake that was found to be heavily polluted with PFAS because it was actually nearby a military base where they used a lot of firefighting foam. This is Hoosick Falls. This was in the news lately. Previously known as New York State's best tasting water, found to have high contamination of PFAS because they were near a fairly significant industrial processing plant. So our plan was to look at radioactive versions of PFAS, to look at routes of bioaccumulation and the kinetics. So we thought, as usually we do, that radio tracers are very useful tools, and that we could radio label our PFAS with fluorine-18. So again, instead of labeling our sugar molecules, we're labeling these environmental contaminants. And so we did a study where we labeled these 3 different compounds. So we call buta, hexa, and octa, PFBA, PFHXA, PFOA, and injected them in mice to look at the different clearance kinetics. So Jennifer Burkemper, who is a chemist in my group, developed this chemistry. So you have your fluorine-18 separation from the target material. She dries it down because this chemistry only goes in very anhydrous conditions, and then did some radio labeling. So we're essentially able to switch out some of these fluorines that are attached to this molecule for our fluorine-18 radioactive atom. Then we have a tracer that we can use for these compounds. So we looked at the kinetics in mice, and we actually found a lot of differences between these families of compounds. We were able to see differences in the blood, differences in the lung, liver, and also, most interestingly, difference in brain uptake. So even very small changes in the structure of these compounds led to fairly dramatic changes in the biodistribution. We also looked at blood binding, and we're able to see very different behaviors of these compounds with the blood binding in these animals. And so it's really interesting. We found these new tools that we can use to sort of monitor these environmental contaminations. So we have this new tool to study the biological distribution of PFAS, and we're starting to look at different families of these PFAS structures. We also had some thoughts that we could use them in environmental remediation studies. So the idea is imagine if you have some water that's contaminated with PFAS and you wanted to know, how are you going to clean up the water? Well, we could take water samples and spike them with our radioactive materials and put them through different filters, do different treatment strategies, and assess quantitatively how well we were cleaning up those water samples. So we're working on these molecular imaging agents. So the primary focus is in medicine. We're also doing some of these studies in the environmental space, but we continue to expand our toolbox. And so I talked a little bit about fluorine. We talked about zirconium, but we're also interested in manganese, cobalt, yttrium, and so we're continually working on new radioisotopes for our new tracers. So one of my postdocs, Lauren Radford, has been working on cobalt-55 production. This is by a PN reaction on nickel-58. So what she's working on is essentially electroplating our nickel-58 onto these coins so we can irradiate those coin targets to make our cobalt-55. Then she's working on separation strategies to separate our cobalt-55 from our nickel-58. And this is just showing the electroplating system. So we have our gold coin that sits on the bottom here, and then we have our anode rod that's used to electroplate our nickel-58 onto that gold coin. We're working on radiochemistry of this folic acid targeting agent. So this is a collaboration with a group in Switzerland. And so we're starting the chemistry, and then we'll start some cell work to look at the binding capabilities and the imaging quality of our new cobalt-55 imaging agent. We're also working on manganese-52. This is an interesting PET analog for manganese MRI agents. Manganese plays important biological roles in both plants and animals, and we're also very interested in mechanism of manganese toxicity, which is known as manganism. So in certain types of industries, namely in welders or manufacturing, people that are exposed to high concentrations of manganese can actually accumulate manganese in the brain. And so here, this is actually a welder that had been exposed to high concentrations of manganese, and you can actually see this bright spot. So this bright spot is actually natural manganese that this patient was exposed to that is picked up on the MRI. So we're looking at making manganese by a PN reaction on chromium-52. This is a PET imaging agent, so again, we're looking at some imaging studies, and we're looking at the ability to image this quantitatively and comparing the images that we get to other isotopes. So we have fluorine-18, copper-64, yttrium-86, zirconium-89, and then we have our manganese-52 agent. And so you can see that the manganese agent you can get very clear and crisp images. So we're very excited to use these manganese compounds in further imaging studies. We're looking at this production by the chromium-52 PN reaction. So we figured out certain methods to make chromium foils. So we use these chromium foils to do the target development. We've looked at yield measurements. So this is just showing the yield of manganese-52. This is the probability that that nuclear reaction is going to happen. And then we've done a lot of separation studies looking at the best way to separate our manganese-52 from that chromium target material. We're also looking at how to electroplate the chromium target material, so we can do this in a more routine fashion. Adriana Massicano, who is one of my postdocs again, has worked a lot on how to electroplate enriched chromium onto these copper coins. And then we've done some very preliminary studies, again, looking at the biodistribution of manganese, the imaging quality, and then we'll start looking at different manganese complexes and how they behave. And so we're really excited to move forward in these new directions. So I thank everybody for their time, and I hope I've managed to convince you that radioisotopes, they play a really important role in medicine and in other areas of science. There's a wide variety of half-lives, there's good imaging characteristics and new chemistries that can lead to a unique toolbox for the development of new nuclear medicine imaging agents and also therapeutics. And of course, development and use of these agents requires a lot of collaboration, both between chemists, physicists, biologists, and physicians. So, and here's our group who does a lot of the work. This is my email if people are interested, and I'm very happy to answer any further questions. Great, thank you.
Thank you so much. Can you unshare the slides?
Sure.
Yeah, we have a few more minutes, 6-7 minutes. So I have a few questions about like how long does it take to make, like, start from the scratch? Like, for example, manganese you started recently. So how long does it take to get the whole thing ready to start some clinical test, or like even in the labs?
So in the lab, so we usually start out with preparation of the targets, and it depends on the compound that we're using as a target material. And so Adriana has been working a lot on the target material, so maybe that took a couple weeks. Then we do some very low level irradiation, so we make very small amounts of manganese. We practice our separation chemistry, and then we start working on the chemistry. And again, it depends how complicated each step is along the way. We do the separation chemistry, then we try to conjugate it to certain targeting agents. And so, you know, these projects with these isotopes can be years long, definitely.
Okay. And like do you collaborate with other I mean, like, universities where you can, I mean, like, this is what I see is a useful research tool to do so many things, like so how, like, have you been looking into collaboration with other universities, other
Definitely,
people?
we have a lot of collaborations. So the
Okay.
that work on the PFAS work, that's a collaboration with University of Notre Dame. We
Okay.
uh, we continue to collaborate with Washington University for the Zirconium-89 trastuzumab. So yeah, no, we're a very collaborative group, and we also welcome. We have collaborations both on people that are interested in making isotopes, and with people that just have new questions that we can probe with our tools. So we will also, I didn't talk a lot about this, but we also are a center for shipping isotopes. So let's say somebody else is really interested in using zirconium-89 for imaging. We can actually manufacture zirconium-89, ship it to them so that we can support their chemistry as well.
And like since it remains in the body for 3-4 days, so have you looked at the toxicity of the radioisotope or anything, or are they
Sure.
very trace levels?
No, that's a very good question. So we get very, very low amounts of radioactivity. That's the very nice thing about PET imaging is it's very, very high sensitivity. So you're really giving tracer amounts, very, very low amounts. There's no biological effect from these. The amount that you're getting was probably on par with having a CT scan. So if you've ever had a CT scan for your knee or head or had an injury or something, that's the kind of levels of radiation that we're talking about.
Okay, and with the patients, like you inject them, like it doesn't have any side effects or anything like that.
That's what I mean. It's the same. The amount of radiation
Yeah,
they're getting
same
is
thing.
very, very
Very
low.
low.
It's like having an X-ray.
Okay, and have you thought of this is kind of like starting something business, because this I see there is a big potential for it to be
So
a
there
big
are, I mean,
business.
so there are businesses. There are
Okay.
commercial businesses that do make FDG or make that sugar analog. And so usually once something's been FDA approved, then and we're using it in a lot of patients, then the businesses will get involved. At the research stage, I mean, we're a university, so that's where we sit, and that's where my heart is too, is in the research side.
Okay. Uh, I think I don't have any more questions right now, and I take this opportunity to thank you for your time and effort. It's really very nice to like. I learned a lot of things. Like I mean, like
Oh,
my my brain is really thinking, oh, okay, we could have done this if at all we had these kind of resources when I
yeah,
was
it's
doing
very
all
big
my
in
stuff.
neuro.
Yeah, yeah. I mean, like especially you are talking about all these glutamate receptors and all those things. So there is a lot of work going on, and with your tool, I mean, like that's a lot of important things could be achieved.
Yeah, a lot of work has been going on
Again,
with Alzheimer's disease and imaging of
other,
amyloid deposition, that sort of thing too.
okay, okay. Again, thank you so much. I really appreciate your time, and good luck with all the things you are doing.
Thank you. Nice to meet you.